7 miles down, pressure that would crush your lungs to the size of ping pong balls, total darkness, freezing cold, zero sunlight. The Mariana Trench in the western Pacific Ocean is deeper than Mount Everest is tall. Challenger Deep, the bottom, sits at 36,000 feet below the surface. You could fit the entire mountain inside and still have 7,000 feet of water above it. Most people think nothing lives down there. They are wrong. The trench is a crescent-shaped scar in the Earth's crust, formed where the Pacific Plate slides under the Mariana Plate. It stretches 1,580 miles long, 43 miles wide. The water pressure at the bottom reaches 8 tons per square inch, more than 1,000 times the atmospheric pressure at sea level. In 1960, Jacques Piccard and Don Walsh became the first humans to reach Challenger Deep in the bathyscaphe Trieste. They spent 20 minutes on the bottom, saw nothing but silt. In 2012, filmmaker James Cameron made the first solo dive. He called it Desolate, Alien, a lunar landscape. For decades, scientists assumed the Hadal Zone, depths below 6,000 meters, was mostly lifeless. They were spectacularly wrong. Chinese researchers, using the Fendosia submersible, which means striver, completed 23 dives into the Mariana Trench in 2024. What they found rewrote the rules. At 9,533 meters depth, almost 6 miles down, they discovered thousands of tube worms and bivalves thriving in complete darkness. The deepest and most extensive chemosynthesis-based ecosystems ever documented. Published in Nature Journal on July 30th, 2025, these organisms don't rely on sunlight. They feed on chemicals, methane and hydrogen sulfide seeping through cracks in the seafloor. Microbes convert these chemicals into energy. The larger animals eat the microbes or each other. An entire food web independent of the sun, life at the absolute edge of what Earth allows. The animals down there are not like anything you have seen. Transparent heads, bodies filled with gel instead of muscle, armor made from aluminum, eyes that glow green in pitch black water, single cells the size of your fist, fish with no scales that can swallow prey larger than themselves. Evolution at the extremes produces creatures that look designed by science fiction writers, but every one of them is real, every adaptation documented, every survival mechanism tested against pressure that would obliterate us. If you value documented deep sea biology and extreme environment research like this, subscribe. It is the best way to support our work. Also comment from which city you are hearing this from. This script is researched and factual. Using sources from peer-reviewed journals, including Nature Volume 2025, published July 30th Grewers, the Scripps Institution of Oceanography Mariana Trench Expedition reports from 2011. PLOS. One journal, April 4th, 2019, on aluminum adaptation mechanisms in Hadal. Amphipods captured from Challenger Deep coordinates, 11 degrees 22.11, north 142 degrees 25.86 east at 10,897 meters depth. And the University of Hawaii Hades expedition findings from 2014, documenting new snailfish species at 8,143 meters. Now back to the information. Number 1, the Mariana snailfish, Pseudoliparis swirei, discovered 2017, the deepest living fish ever recorded. 8,076 meters down in the Mariana Trench, almost 5 miles below the surface, researchers using baited camera traps spotted something moving. White, translucent, broad wing like fins, an eel-shaped tail. Gliding slowly over the seafloor, they filmed it multiple times. Then they caught 37 specimens between November 15th and November 25th, 2014, using mackerel-baited traps. The holotype specimen, catalog number USM 438975I, was caught November 21st, 2014 at 7,949 meters depth. The fish has no scales, transparent skin. You can see through its body to the internal organs. Certain organs are enlarged to handle the pressure. The eggs are bigger than in shallow water snailfish. The muscles are thinner. The bones incomplete. The skull is barely ossified, soft, flexible. This lets the internal and external pressures equalize. At that depth, the pressure reaches 15,750 pounds per square inch, enough to compact a human body into a dense flesh chunk. The snailfish survives because its body is mostly water and gel, like tissues that can't be compressed. It has little to no vision. Eyes don't work in total darkness under crushing pressure. Instead, it hunts by feel and chemical detection. The fish feeds on tiny crustaceans, amphipods. It clusters in small groups, drifting along the trench floor, picking off prey that ventures too close. Scientists caught specimens ranging from 22,600 feet to 26,200 feet deep. depths where most fish would implode. The species is named for Herbert Swire, first navigating sub-lieutenant of HMS Challenger. In 1876, the Challenger expedition discovered Challenger Deep. 148 years later, we found the fish living there. Studies show 8,000 to 8,500 meters appears to be the theoretical maximum depth possible for fish. Below that, proteins stop functioning. Cell membranes lose flexibility. The Mariana snailfish lives right at that edge. Later expeditions in 2023 filmed another snailfish species, still unidentified, at 8,336 meters in the Izu Ogasawara Trench off Japan. The deepest fish ever filmed. But the Mariana snailfish holds the capture record, 7,949 meters. The fish pulled from the deepest documented depth. The body looks different from other deep-sea fish. That white translucent appearance is unique. The pectoral fins are enormous, wing-like. They use them to hover motionless in the water, conserving energy in an environment where food is scarce. Analysis of stomach contents shows they eat hadal amphipods, the same crustaceans that dominate the trench floor. One specimen's last meal was visible in a CT scan, green mass in the digestive tract. Amphipod remains. The fish is perfectly adapted to life where nothing else with a backbone can go. Number 2, the supergiant amphipod, Allicella gigantea, the largest amphipod on Earth. Most amphipods are 2 to 3 centimeters long, about an inch. This one grows to 340 millimeters, 13 inches, over 10 times bigger than its relatives. In November 2011, researchers deploying camera traps and baited landers in the Kermadec Trench north of New Zealand pulled up something impossible. 7 specimens, white, segmented, massive. The largest measured 290 millimeters, 11 inches. Cameras on the seafloor photographed one estimated at 340 millimeters, more than a foot long. Crustaceans that size should not exist at 7,000 meters depth, but they do. The species was first described in 1899 from specimens caught in the Madeira abyssal plain off the Canary Islands, then nothing for decades. Rare sightings in the 1970s off Hawaii, then silence. Scientists assumed it was extinct or incredibly rare. In 2012, the Kermadec Trench expedition proved otherwise. Not rare, just hard to find. Recent research published in Royal Society, Open Science in 2025, compiled 195 records from 75 locations worldwide. Pacific Ocean, Atlantic Ocean, Indian Ocean. Depth range, 3,890 to 8,931 meters. DNA analysis from specimens collected from 9 trenches and 3 fracture zones confirmed it is a single globally distributed species. Habitat modeling suggests Allicella gigantea may inhabit 59% of the world's oceans, anywhere between 3,890 and 8,931 meters deep. That is over half the planet's ocean volume. Not rare at all. Just living where we almost never look. The supergiant is white, no red pigment. Red wavelengths of light get absorbed quickly by water and don't reach the deep sea. Most deep sea amphipods are red or orange to avoid predators. But Allicella gigantea is white because it has no predators at those depths. It is the apex scavenger. When something dies and sinks to the trench floor, the supergiant shows up to feed. They detect carrion from great distances using chemical sensors. Baited traps draw them in within hours. Mackerel carcasses, dead fish, anything organic, they swarm it. The species has an exceptionally large genome, 34.79 gigabase pairs, considerably larger than other deep amphipods. Scientists hypothesize this relates to its gigantism. Genetic studies from 2021 found genes related to growth regulation are overrepresented. The cold temperature and extreme pressure at depth may increase cell size and lifespan. Larger cells, longer life, bigger body. Carbon-14 dating of tissue samples suggests these amphipods live over 10 years. Slow metabolism, extended lifespan, an adaptation to food scarcity. Females brood eggs in a pouch. Eggs measure 6.95 to 14.88 millimeters long. Oval-shaped, 2-layered shell. As juveniles, they eat bacteria and zooplankton debris. As adults, they switch to carrion and algae. Pure scavengers. The team that caught them in the Kermadec Trench deployed equipment on the same site days later. No supergiants appeared. They were there one day and gone the next. Nomadic, following food sources across vast abyssal plains. Specimens caught weighed up to 138 grams in the Kermadec Trench, a single amphipod heavier than 5 ounces. Impossibly large for a creature that should be thumb-sized. Number 3. The aluminum-armored amphipod. Hirundellea gigas, the deepest living amphipod ever recorded. 10,897 meters. Challenger Deep, the bottom of the Mariana Trench. Researchers captured specimens of Hirandelia gigas at coordinates 11 degrees 22.11 north, 142 degrees 25.86 east. Analysis of the exoskeleton showed something unprecedented: aluminum. Published in PLOS One Journal, April 4, 2019. First documented case of any crustacean using aluminum for structural protection. The amphipod's exoskeleton contains primarily calcium carbonate, standard crustacean armor, but coating the surface is aluminum. Widely distributed across the entire body, especially high concentrations on the tail, the telson, and along the edges of the feet, the periopods and uropods. Electron microscopy showed the aluminum sits on the surface, not accumulated internally, a protective gel layer. When researchers washed specimens with distilled water, the aluminum came off. It is an external shield. Here's how it works. Aluminum is rare in seawater but abundant in sediment. Hirandelia gigas produces gluconic acid and gluconolactone in its gut. These organic compounds extract aluminum from the seafloor sediment the amphipod ingests. The extracted aluminum ions then react with the alkaline seawater to form aluminum hydroxide gel. This gel coats the exoskeleton. Pressure experiments conducted at 100 megapascals, equivalent to 10,000 meters depth, showed the aluminum gel protects the calcium carbonate shell from dissolving under extreme pressure. Without the aluminum, the exoskeleton degrades. With it, structural integrity holds. No other amphipod does this. Researchers analyzed a shallow water coastal amphipod species, Prontogenesia, from Maizuru Bay in Japan. No aluminum present. This is a Hadal zone adaptation, specific to the deepest living crustaceans. Hirandelia gigas uses aluminum to survive, where calcium carbonate alone would fail. The discovery came from analyzing 6 individuals out of 185 captured from Challenger Deep. All 6 had aluminum. 3 more individuals from the Izu Ogasawara Trench at 9,450 meters also showed aluminum in their exoskeletons. The species ranges from 7,000 to nearly 11,000 meters depth. It has been recorded at 10,908 meters in multiple trenches. Mariana, Kermadec, Izu Ogasawara, New Britain. They feed on wooden debris, sunken logs, ships, anything organic that reaches the bottom. Hirandelia gigas has evolved cellulase enzymes capable of digesting wood, unique among deep sea animals. When nothing else is available, they eat the dead trees that occasionally sink into the abyss. The aluminum shield lets them survive long enough to exploit resources nothing else can touch. Number 4. The giant single-celled amoeba, Xenophytophores, 4 inches across, 1 cell, July 2011, Scripps Institution of Oceanography, expedition to the Mariana Trench. Researchers deployed drop cams, baited camera landers that free fall to the seafloor and film for hours before sending. At 10,641 meters depth, 6.6 miles down in the Serena Deep, they spotted something bizarre. Pale lumps on the sediment, sponge-like. Massive, 4 inches in diameter, 10 centimeters, single-celled organisms, the largest individual cells known to exist, Xenophytophores. The name means bearer of foreign bodies. They construct elaborate tests, shells, from sediment particles glued together with organic cements. Calcium carbonate, sponge spicules, radiolarian tests, even smaller foraminifera shells. They extract minerals from their surroundings and cement them into protective structures. Some are soft and lumpy, others form fans or complex branching patterns. The tests can reach 20 centimeters across, 8 inches, 1 cell commanding a structure the size of a dinner plate. The protoplasm contributes less than 1% of the organism's total mass. Most of the xenophyophore is its constructed shell and waste material. They have many nuclei. Technically, they are multinucleate unicellular organisms, a type of foraminiferan. They feed by trapping particles from the water. Bacteria, organic matter, they concentrate heavy metals, lead, uranium, mercury. Studies show they are highly resistant to toxic doses. An adaptation to sediment rich in chemicals. The previous depth record for Xenophytophores was 7,500 meters in the New Hebrides Trench. The Mariana sighting broke that by over 3,000 meters. These organisms are exclusively deep sea, found from 500 meters down to over 10,000. They are abundant on abyssal plains. In some regions, they are the dominant species. 75 species described so far. 15 genera. The largest, Syringamina fragilissima, reaches 20 centimeters. The biggest, Coenocyte, a mass of cytoplasm containing multiple nuclei ever documented. Research by Lisa Levin at Scripps Center for Marine Biodiversity and Conservation confirmed the identifications. Andrew Gooday of the UK National Oceanography Centre verified them. The organisms play host to diverse multicellular animals, small worms, crustaceans. They provide habitat in an environment with almost no structure. The dropcam footage showed them scattered across the seafloor, pale blobs, fragile. If you tried to collect one, it would fall apart. That is why they are poorly studied. Too delicate to survive being hauled to the surface, but at depth they thrive. Single cells dominating the Hadal ecosystem. Number 5, the transparent headed fish, barreli, Macropinna microstoma, eyes that look through the skull. 2,000 to 2,600 feet deep, 610 to 795 meters. The barreli fish hovers in the midnight zone of the Pacific Ocean, depths where sunlight fades to nothing. The fish has a clear dome on its head, a fluid-filled transparent shield. Inside, 2 bright green tubular eyes point straight up, barrel-shaped, aimed at the surface. The fish can see through its own skull. The adaptation maximizes light detection in near total darkness. The tubular eyes are highly sensitive. They spot the faintest silhouettes of prey against the dim background light filtering from above. The green pigment may filter out surface light wavelengths and enhance detection of bioluminescence, jellyfish, siphonophores, other glowing creatures of the deep. The barreli steals food from the tentacles of siphonophores. It uses its tiny, precise mouth to pluck zooplankton from the stinging tentacles without getting hurt. The transparent head protects the eyes during feeding. Scientists did not understand this for decades. The fish was first discovered in 1939, pulled from 2,500 feet depth. For 70 years, researchers thought the eyes were fixed upward, permanent skyward gaze. Then in 2009, the Monterey Bay Aquarium Research Institute observed living barrelets using remotely operated vehicles. The eyes rotate. When the fish spots prey above, eyes point up. When it moves forward to feed, the eyes roll to face forward. A flexible optical system that adapts to hunting behavior. The fish grows to 4 inches long, 10 centimeters. Fully grown barrelets are barely longer than your finger. The body is brown and ordinary looking below the transparent dome. 2 indentations where you might expect eyes are actually olfactory organs, smell detectors. The real eyes sit inside the cockpit of the head. The large, flat pectoral fins let the fish remain suspended motionless in the water column, hovering, waiting. Minimal energy expenditure. When prey appears, quick, precise movements, then back to hovering. The species ranges across the Pacific, between Japan and Baja California. Bering Strait, deep waters far from shore, they feed on zooplankton, microscopic animals, plentiful in the ocean. The barreli's elongated intestine and multiple ceca, lateral dilations of the gut, show specialization for digesting plankton. Most plankton don't live as deep as the barreli, but siphonophores trap plankton in their tentacles and transport it to deeper waters. The barreli follows, parasitizing the siphonophores' catch, a survival strategy built on theft. Number 6. The deep-sea dragonfish, fangs in the dark, bioluminescent predator, 700 to 6,000 feet depth, 213 to 1,830 meters. The dragonfish patrols the Mariana Trench's upper reaches, scaleless body, eel-like skin, black as ink. Teeth that are grotesquely oversized. The fangs are so large the fish cannot close its mouth. They protrude even when the jaw is relaxed. This is a predator designed for one thing: killing. The fish uses bioluminescence for hunting and communication. Light-producing organs called photophores cover the body. The dragonfish flashes, patterns, attracts prey, signals to mates. Some species have evolved the rare ability to produce red bioluminescence. Most deep-sea creatures cannot see red light. It is invisible to them. The dragonfish uses red light like a sniper's infrared scope. It can see prey that cannot see it, illuminating victims without alerting them. Then the strike, jaws snap shut, fangs impale. The prey dies before it knows the dragonfish was there. Dragonfish grow to about 6 inches, 15 centimeters, small body, huge teeth. The fangs are the largest teeth relative to body size of any fish in the ocean. They feed on other fish and squid, ambush predators, hanging motionless in the water column until something swims close, then explosive acceleration. The hinged jaw opens wide, far wider than seems possible. The fish can swallow prey larger than its own head. The species survives in an environment with almost no food. Meals are rare. When one appears, the dragonfish cannot afford to miss. The oversized teeth ensure the prey cannot escape once bitten. Backward-facing barbs. The more the victim struggles, the deeper the fangs penetrate, fatal every time. After feeding, the dragonfish can go weeks without eating. Slow metabolism, low energy expenditure, floating in the dark, waiting for the next meal. The scaleless skin is an adaptation to reduce drag, smooth movement through water, less energy required. Every calorie matters at depth. The dragonfish is perfectly engineered for efficiency, minimum input, maximum lethality. A living weapon shaped by the most hostile environment on the planet. Number 7. Chemosynthetic tube worms and bivalves. Life at 9,533 meters, 2024. The Chinese submersible Fendosia, Striver, dove 23 times into the Mariana Trench. The deepest and most extensive biological survey ever conducted. At 9,533 meters, they found something unprecedented. Thousands of tube worms and bivalves, dense aggregations, entire colonies thriving in complete darkness. The deepest and most extensive chemosynthesis-based communities ever discovered. Published in Nature, July 30th, 2025. These are not small scattered individuals. This is a massive ecosystem. Tube worms clustering around white microbial mats that resemble snow. The mats are methane-producing microbes. The tube worms position themselves over the mats and absorb chemicals, hydrogen sulfide, methane, bacteria in the tube worms. Bodies convert these chemicals into energy, chemosynthesis. The tube worms have no mouth, no digestive system. They survive entirely on the bacteria living inside them. Symbiosis at the cellular level. The bivalves, clams, are equally adapted. Dense beds at 5,743 meters at a site called Clam Bed. The species is Archivesica phaseoliformis. They burrow into sediment and filter feed on bacteria and organic particles. The fluids seeping through the seafloor faults are rich in methane and hydrogen sulfide. These chemicals are produced microbially from deposited organic matter. Isotopic analysis confirmed it. The carbon signature matches microbial methane production. The discovery rewrites models of deep ocean carbon cycling. Scientists assumed chemosynthetic communities were limited to hydrothermal vents, hot water vents along mid-ocean ridges. Those typically occur at 2,000 to 3,000 meters depth. Finding thriving chemosynthetic life at nearly 10,000 meters means the deep biosphere is far more extensive than previously recognized. Other trenches likely have similar communities. Kermadec, Tonga, Japan, Peru, Chile, anywhere with tectonic activity and methane seeps. The researchers also found compelling evidence of echite, a rare calcium carbonate mineral that forms only in cold seep environments. Samples recovered from the cold seeps showed echite crystals. Photomicrographs confirmed the structure. Scale bar, 1 centimeter. This mineral indicates active chemical seepage. The communities are sustained by continuous flow of methane-rich fluids transported along faults traversing deep sediment layers. The team included researchers from multiple Chinese institutions. Lead author Xiaotong Peng published findings in Nature, volume 635. The paper stated these chemosynthesis-based communities might be more widespread than previously anticipated. Given that other ocean trenches are similar, the Hadal Zone could harbor extensive life. Not desolate, not barren, alive, teeming, a frontier ecosystem feeding on Earth's chemical exhaust. Deep sea mining threatens these communities. China, the United States, and other nations have expressed interest in mining the seafloor for valuable minerals. Cobalt, nickel, rare earth elements. The International Seabed Authority, which oversees deep sea mining in international waters, has not yet adopted rules governing the industry. Scientists warn that mining could decimate fragile ecosystems not yet understood, destroying life before we even catalog it. The tube worm and bivalve colonies discovered in 2024 underscore the urgency. We are only beginning to understand what lives down there. These 7 animals represent the absolute extremes of life on Earth. The Mariana snailfish at 8,076 meters, the supergiant amphipod ranging across 59% of the deep ocean, the aluminum-armored Hirandelia gigas at the bottom of Challenger Deep, the giant xenophophores, single cells as large as your fist, the barreleye with its transparent head and rotating eyes, the dragonfish with fangs that never retract, and the chemosynthetic colonies at 9,533 meters, proving life thrives where sunlight never reaches. Each one defies what we thought possible. Transparent heads, aluminum shields, single cells 10 centimeters wide, fish living at depths that should be sterile, ecosystems built on poison gases. The Mariana Trench is not a dead zone. It is an alien world, 7 miles down, right here on Earth. In 2025, a soil core analysis from the Clam Bed site at 5,743 meters revealed sediment layers rich in organic carbon. The carbon dates to surface origin, material from the photic zone, dead plankton, decomposed algae, marine snow. It drifts down through miles of water and settles on the trench floor. The tube worms and bivalves consume it. A direct connection between surface life and hadal life. What dies in the sunlit ocean feeds the abyss. The food web extends from 0 meters to 10,000 meters, top to bottom. The entire ocean column is interconnected. Pollution follows the same path. Microplastics have been found in hadal amphipod stomachs, PCBs, and snailfish tissue. Even at the bottom of the world, human impact is measurable. The aluminum gel on Hirundellea giga's exoskeletons represents 138 years of evolution we are only now discovering. Since the Challenger expedition in 1876 found the trench, life has been down there adapting. Building armor from seafloor sediment, extracting metals, coating bodies with chemical shields. We sent a bathyscaphe down once in 1960 and saw nothing, declared it barren. In 2012, Cameron went down and called it desolate, but in 2024, Fenduzi found thousands of animals, colonies, communities. The hadal zone is not empty, it is full. We just were not looking in the right places with the right tools. The Mariana snailfish specimen, 7,949 meters deep. Catalog USNM 438975 ops, sits preserved in a lab in Washington. Tissue samples reveal genetic adaptations. Proteins stabilized against pressure. Cell membranes modified with unsaturated fats to maintain flexibility. Enzymes that function at near freezing temperatures under crushing force. The fish's genome holds answers to questions we have not yet asked. How does life persist at the edge of physical possibility? The answers are written in the DNA of creatures living where we cannot go. The supergiant amphipod specimen, 290 millimeters long from the Kermadec Trench at 7,000 meters, resides in Wellington, New Zealand. White, segmented, impossibly large. Researchers measured its genome, 34.79 gigabase pairs. Over 10 times bigger than the human genome, junk DNA, transposons, repetitive elements, or perhaps regulatory sequences we do not yet understand. The gigantism is encoded in there, somewhere. The mechanism by which a 2-centimeter amphipod becomes a 34-centimeter supergiant. Cold, pressure, food scarcity, time. Evolution solved the problem. We are still catching up. Xenophyophore specimens from 10,641 meters in Sirena Deep shatter our assumptions about cell size. Biology. Textbooks say cells cannot grow beyond a certain size. Surface area to volume ratio. Nutrient diffusion limits. Structural integrity fails. But the xenophyophore ignores the rules. 4 inches, 1 cell. It works because most of the organism is not living tissue. It is scaffolding, mineral test, waste products. The living cytoplasm occupies thin threads running through the structure. Nuclei scattered throughout. It is a biological city, 1 cell sprawling across centimeters. The trench is 5 times longer than the Grand Canyon, much deeper, 1,580 miles, 43 miles wide, 10,984 meters at the deepest point, enough water to cover Mount Everest with 7,000 feet to spare. The volume of the hadal zone, depths below 6,000 meters, represents less than 2% of the ocean. But that 2% contains biodiversity. We are only beginning to catalog. 15 genera of xenophyophores, 75 species, over 100 amphipod species, dozens of snailfish varieties, jellyfish, sea cucumbers, isopods, countless microbes, bacteria, archaea, viruses. Cameron called it desolate because he landed on silt, barren seafloor. But life in the hadal zone is patchy, clustered. You can descend 7 miles and see nothing, or you can hit a cold seep and find thousands of tube worms. It depends where you land. The Fenduzi made 23 dives, systematic survey, covering distance, mapping communities. That is how you find life at depth, not 1 dive, dozens, persistence. The trench is vast. Life is concentrated where resources exist. Methane seeps, sunken wood, carcasses, animal falls, whale skeletons. 1 gray whale sinking to the trench floor provides food for decades. Scavengers arrive first, amphipods, isopods, fish. They strip the soft tissue in weeks. Then bone specialists move in, ossidax worms, bone-eating snot flowers. They drill into the skeleton and extract lipids. The whale's bones sustain an entire ecosystem for 50 years. Chemosynthetic bacteria colonize the decomposing marrow. A whale fall creates an oasis, life radiating outward from death. In the hadal zone, every calorie is precious. Nothing is wasted. The bivalves at clam bed filter liters of water per day, extracting bacteria, organic particles. They build shells from calcium carbonate. The shells accumulate, layers of sediment mixed with shells. Over time, this creates structure, Habitat. Small worms burrow into the shell hash. Crustaceans hide in crevices. One species supports others. Ecosystem engineering. In an environment with no hard substrate, the bivalves create it. They change the trench floor, make it livable for others. The dragonfish hunting at 1,830 meters sees the trench's edges. Slopes, dropping into darkness. It never ventures deeper. The pressure at 6,000 feet is manageable. Below that, the fish's physiology fails. Swim bladder collapses. Scales crack. Proteins denature. Each species has a depth limit. The dragonfish stops at 1,830 meters. The barreleye at 795. The snailfish reaches 8,076. The aluminum armored amphipod hits 10,897. Below that, only bacteria and archaea, single-celled extremophils. The absolute limit. The Mariana Trench continues to reveal secrets. Every expedition finds new species, new behaviors, new adaptations. The Hadal Zone is Earth's final frontier. Deeper than space is high. We have sent 12 people to the moon. Only 27 have reached Challenger Deep. We know more about the lunar surface than the trench floor, but that is changing. Submersibles improve. Landers multiply. Cameras advance. Genetic analysis accelerates. The next decade will catalog more Hadal life than the previous century. The 7 animals here are not the strangest the trench has to offer. They are the ones we have documented, filmed, captured, analyzed, published, peer-reviewed, confirmed. How many species remain undiscovered? How many unique adaptations? How many survival strategies? The trench is 1,580 miles long. We have surveyed fractions of a percent, tiny slices, specific sites. The rest is unexplored. Challenger Deep, Serena Deep, Clambed, a handful of locations. The rest is darkness. The aluminum shield on Hirandelia gigas was discovered in 2019. We have been studying deep sea amphipods for over a century. But only when researchers applied electron microscopy to specimens from 10,897 meters did they see the aluminum. It was always there, coating the exoskeleton. We just were not looking for it. How many other adaptations are we missing? The xenophyophories concentrate uranium, lead, mercury. In quantities that would kill most cells, they are bioaccumulators, metal sponges, potential biomonitors for pollution, or sources of novel compounds, pharmaceuticals, industrial applications. We do not know yet. The chemosynthetic tube worms at 9,533 meters have bacteria we have never cultured. Novel species, unique metabolic pathways. They convert methane and hydrogen sulfide into energy. No sunlight, no photosynthesis, pure chemistry. If we understood the mechanism, we could replicate it. Artificial chemosynthesis, energy production from waste gases, industrial applications, climate solutions. The biology is there, proven, working. We just need to decode it. Trench animals are not curiosities. They are blueprints, survival strategies tested against extremes, pressure, cold, darkness, food scarcity. Each adaptation is a solution. Engineering problems solved by evolution. Transparent heads to maximize light detection, aluminum coatings to protect shells. Giant genomes to regulate extreme growth, single cells expanding beyond normal limits. Each one a lesson, each one applicable. The barrelized tubular eyes inspired optical designs, rotating lenses, adjustable fields of view. The dragonfish's red bioluminescence led to stealth lighting research, invisible illumination. The snailfish's gel body inspired pressure, resistant materials, flexible, incompressible, adaptable. The principles transfer, biomimicry. Nature solved these problems millions of years ago. We are learning from the solutions. The Mariana Trench is 7 miles down. The animals there are unreal, but they are real. Everyone documented, every adaptation verified, every survival mechanism proven. The Hadal Zone is not empty. It is full. Full of life engineered for the impossible, full of creatures that should not exist, but do. The Chinese expedition that found the tube worm colonies in 2024 continues. More dives planned, more sites surveyed. The Fenduja will return. Other submersibles will follow. The Hadal Zone is opening. The secrets are emerging, one dive at a time, one discovery at a time. The abyss is not giving up its mysteries easily, but we are pulling them out, species by species, adaptation by adaptation. The deepest ocean is becoming known. 7 animals. 7 extremes. 7 impossibilities proven possible. The Mariana Trench holds them all, and more. Waiting. 7 miles down, in darkness, under pressure, alive.